I promised a recording on the rest of Chapter 2. I apologize, I'm a little stuffy, I caught a cold. So, there are 4 important protein families in cell-cell adhesion and creating tissue. We've talked about integrins already. We talked a lot about that on Friday. So our other family members are selectins, immunoglobulins, and cadherins. So immunoglobulins are also what we think of when we think about immune cells and creating antibodies. Selectins and cadherins may or may not be regulated by calcium. So these are calcium dependent. And then our adherents. So our cell-cell adhesion molecules can either function in a homophilic, so they bind one cell to the other cell by binding to the same type, so a cadherin here and a cadherin here, or they can be in a heterophilic manner, so a selectin here can bind to a different protein on the other cell, or an integrin on this cell is going to bind to fibronectin, and then that binds to the extracellular matrix. So there are lots of different ways to form that Velcro between one cell and the other cell. So cadherins are homophilic. The immunoglobulins can be either or, so they can be, you can have one, it's shortened by CAM, so it can be one CAM to another CAM, or it can be an integrin to a CAM. We talked about integrins, and then selectins bind to mucinin or other glycoproteins on the other cell. So let's go one by one. First one are selectins. You can have a couple different kinds of selectins, so L-selectin, E-selectin, P-selectin. So the first thing you notice, there's different links. So you have different links of these structural domains. They have, then on the L, they have this lectin type like domain, so that just means they like to bind sugars. So here you can see on that other cell, they're going to bind sugars, whatever sugar there is on a glycoprotein. So you have sialic acid, then a couple other sugar moieties on the end of this proteins. So once the one they show on that, what I showed on that previous slide was mucinin. E-selectin, this one here is going to likely be on endothelial cells. P-selectin can be on platelets and endothelial cells, and L-selectin is something important on a white blood cell or a leukocyte. And this is L-selectin is going to be important when you think about white blood cells trying to get to sites of inflammation. So our immunoglobulin superfamily, these proteins can bind to one another, so homophilic, or they can bind to heterophilic, so they could actually bind to our integrins. But they're all going to have this IgG domain, so this back and forth characteristic IgG domain. They have immune purposes, but they also are important in keeping cells stuck together. We have our vascular cell adhesion, neuronal cell adhesion, and neuronal development. So we can kind of deduce from their name what their purpose might be. Our cadherins, they're glycoproteins that are going to mediate calcium-dependent cell adhesion, and they kind of work like they zip things together. So they're going to first bind end to end, and then pull themselves closer and closer. They're going to pull those two plasma membranes closer and closer. We have at least 30 different glycoproteins, and they kind of get classified in these three bins. So E cadherin for epithelial, N for neuronal, and P for placental. And these are quite important for development. So as the cadherins kind of pull themselves closer together, right? So they're all like this. They're going to pull that plasma membrane closer together. As they do that, we can see how, as they pull them together, we could probably pull all these cells closer together. So here I could pull, I have my two cadherins, one here and one here. If that got closer together, I could pull those cells together. And that's what's really important in forming that neuronal tube during development. So our cell junctions, adherens junctions, desmosomes, tight, gap, and hemidesmosomes. So here are our tight, we're preventing regulating what molecules can get through. Those are basically impermeable except for the things we want to get through. Adherens junctions are these guys here. This is connected to the actin cytoskeleton. We have our desmosomes that are here. Our hemidesmosomes are connected to the basement membrane. Gap junctions allow things to go through each one of these cells. So first, what I said, adherens junctions, we are making this belt around near the apical surface of a cell. And then towards the top, you can see that here. And they're held together by a calcium-dependent linkage. So that calcium-dependent linkage is cadherin. So I have my two cadherins are binding to each other between cell 1 and cell 2. On the other side, so this is inside of cell 1, inside of cell 2. This complex is connected to the actin cytoskeleton. It's connected by, so we have cadherin here, and I have this protein called beta-catenin that connects to the actin cytoskeleton. So actin cytoskeleton is also giving structure to this cell over here. And it can send signals. So if there's calcium changes here, and this decides to let go, it's going to send a signal to the nucleus to turn genes on or turn genes off. So the desmosome, those look very similar, right? That looks very similar. I have these proteins with these multiple domains, right? They bind to each other in a very similar way, right? They're kind of forming this stacked structure. They're connected across the cell membrane to another complex, to these things called intermediate filaments. Intermediate filaments are very similar to actin. They're just made up of different proteins. They give structure to the cell. So this looks very, very similar, right, to an adherens junction. We have different proteins. So this is desmoglein or desmocolin. They kind of give a dish shape, so they're a little differently shaped than an adherens would. So you can kind of see all these. If we go back here, the cadherens aren't quite. I'll go back here. There's not quite as many proteins scattered together tight, so this is more dense. You see a lot more. It's much more dense proteins together. So we have a lot more connection between these desmoglein and desmocolin forming this junction, giving a lot of structure. These are really important in things that have a lot of mechanical stress, so your skin, your gums, because you're putting all these proteins together in a nice tight complex. So our tight junctions, these are the gatekeepers. This is making sure that nothing can get through this unless you want it to. So they're only permeable to specific ions or solids, things you really want to get through. Not going to let any water through unless you say. They're created by interactions between claudins and occludins, so that these are proteins that are transmembrane proteins expressed on the outside of the cell that bind to one another. If these mutations in here, there, or disruption of these interactions are going to make these come apart, and then you're no longer going to have that sealed space. And this is just a nice image of what a tight junction looks like. So what is a gap junction? These are communication. So these are a way for, these are in mammalian cells, this is plant cells. This is a way to communicate between cells and cells. So make a protein and send it from one to the other. This is the secret passageway. It's made up of the protein called connexins, and it's really just a tube to send into the other cell. You can send things that are low molecular weight that's only a certain size. You wouldn't be able to send a huge protein complex from one cell to the other. So there is a cutoff. but it's an excellent way to not have to send something out. You don't have to exocytose something and then endocytose. You can just send it through the cell. So why is this important? I'm going to upload this video right here onto Blackboard. But all this is really important, not only in development, but in the inflammation process. You express on your epithelial cells are going to express selectins on the surface of the cells, right? But you want to have your white blood cells, your neutrophils. You want them to just casually run along. You want them to roll. You want them to only come into your tissue when it's time. You don't want them to actually activate inflammation until there's a reason. So usually on the surface of the white blood cell, on the neutrophil, they don't express the integrin. It's unactivated until the signal has been sent that it should be activated. So they usually just roll along until there's an activation. Then they form an actual type. There's actually a connection between the two, between the P and E selectins. Then there actually is this thing called diapedesis, where they move between the two cells. But usually it's not tight enough. They just tight enough of an interaction. They roll right along. So what do I hope you learned from this chapter? The different types of proteins, the different connections, both between that creates the extracellular matrix, that makes the basement membrane, how you connect cells to cells to one another, and our different types of junctions. I will see you on Tuesday.